Hello, dear students. Today we will talk about ethics of scientific biomedical research. As, this issue is very famous, and nowadays we have um, very high attention to this topic. Um, of course, it's we start from animal experiments because it's like basis for scientific research. It's easiest way to check hypothesis, check of our theory on practice before this knowledge or these approaches testing on humans. Of course, it's human first. Uh, in, Interesting, but in Ukraine we have quite good legislation about animal protection. First document was adopted, approved quarter of this century ago in, on on start of this century. It's in the same day with analogical analogical documents in Strasbourg in 1985, also on 20, by 20th September. Later, in Верховна Rada in Ukraine approve law of protection animals from cruelty. This is article of this law number 26 talk about regulation from animals which used in scientific experiments, testing, education, and this also rising in this document rising question about alternative non-animal methods in uh, some improve some mechanism of uh, interaction between between human and animals and some in this first law in this legislation in Ukraine. Uh, Central mechanism is this is it's a local ethical committee. It's which approve or disapprove these experiments, watching about this need necessity of these experiments, this evaluate of number of animals involved in experiment and the usage of painkillers, analgetics, narcosis, and this some taking these animals in captivity, etc. Um, Interesting in 2012, Minister of Education of Science signed order prohibiting of use animals in labs, university labs. Um, in latest law activity is this is proposal of Ministry of Health uh, in form of draft law about banning testing cosmetics on animals. But this for today, this draft not approved by Ukrainian Parliament, Верховна Rada. Um, in Union. Now it's actual directives from 2010. This about protection animal about protection animals in scientific research, and this directive regulate this activity on this field. Uh, it's about some basis of usage experimental animal. It's the issue of inhuman treatment of these animals during experiment. It's some organization and people who work in animal protection area, animal so called animal defenders demand it's some inhuman treatment of domestic animals, prohibit usage animal in circus and delphinariums, prohibit mass killing of wild animals, and also these demands tied with scientific area, and this we have probably it's about 4 or 5 years ago. It's animal defenders protesting in our medical institute because somebody complains about this some in labs some experiments on with infection disease on these mice. It was wrong information, but in COVID era, a lot of people worried about something like that, this experiment and infection, and this TV and some activists check medical facilities of medical institute because this local society worried about something something criminal. It what were in medical institute animal house. Okay, it's inf- some information, fun information, but nowadays we can find this video from local TV about this subject, this history story. Uh, these questions arise lots of debates because it's scientific progress. It's impossible was impossible without animal experiments. Lots of pharmacology, medicines, drugs. It's tested on animals, and then before it's run these clinical trials in human. Of course, it's obvious. It's it's difficult without this stage trying use something for humans. Uh, okay, because it's a new chemicals. It is impossible to use this for humans without these animals. Um, But is this in this debate rise question about this uh, minimal usage of animal models for these preclinical studies, and use special artificially bred lines of animals. Usually, it's mice or these flyers of drosophiles. Um, last. Uh, In last decades, this introduct new models in these animal experiments, like usage of worms. An example, Cenorhabditis elegans. Uh, by the way, it's nowadays it's still this still active, this obligation of first experiments conducted on lab animals in order to minimize risk of harmful effects on human body. Despite of this all debates. Uh, from historic periodization in historic perspective, it's when this scientific knowledge start development in some very fast. And of course, it is people, scientists, researchers conducting experiments on animals, of course, without anesthesia. First, in second stage in the 19th century, it's public society. What is first voices against animal experimentation, and in 1878 it was approved first law for protection animal experiment animals, and its meaning of this law, it's ordering use of painkillers. Uh, 20th century gave us a lot of experimental science, and of course its most famous convention is European Convention for Protection Vertebrate Animals used for the experimental and other specific purpose from 1985. It's probably first big law. It's or convention, or this first document which uh, this shed this light on this problem very well. In Great Britain, Birch and Russell this create concept of humanization, medico-biological natural science experiment with very well-known principle. Three are humanic human behavior with in experiment with animal. Its first are its replacement. It's maximal usage of alternative materials and methods instead of live animals. An example, it's introduction of tissue culture. It's some fibroblasts, endotheliosites, and epithelial cells. Um, Introduction of microbacterial culture for replacement vertebrates, and also use of computer and mathematical modeling. Next R is reduction. It's minimize number of animals in experiment. It's skip some control groups and unnecessary control. Use its minimal number. It's an example in the first, most of its statistics. It's five animals, and usually researchers take six animals. It's like one animal. It's like in case some disaster with with these animals in the case of some this animal sick, died in some reasons, and this like insurance for some tragical cases with animals. Uh, Refinement. It's improvement of uh, lab technology, lab experiments with animals. It's this activity include minimization of pain, discomfort, inconvenience for experimental animals. Yes, maximum protection of animals during experiment. What in local practice it's for avoid some problem with in publication process in with some funding body of which receive some funding from European Union abroad or from ministry use standard strains which don't need permission. It's use some commercially available cell cultures and also local ethical commission produce some documents for our activity for avoid some problems with legislation and legal scientific practice because without these documents we can't produce some papers with good publication impact factor, etc. It's obligatory for if you want creating something meaningful and something important. uh, it's this regulation nowadays widespread and mandatory in most of countries around the world. There's lots of documents, local documents in different country for this. We should take in account if we have international collaboration and all partners provides these documents mm, for taking they taking in account these ethical principles. Usually, this all these ethical principles coordinate and correlate with European Convention from Strasbourg, 1985. Um, it's in its meaning of this registration and etc. It should follow rules of selecting a right animal model and should staff who work with animals which need some um, response for demands. So it's like it's in staff we should have veterinarian. It is some uh, training. These people should take training courses, and this also animal houses should have some documentation, logbooks, etc. Also, this all experiments supported by ethical committee approval. Um, is this usual alternative methods? It's cell culture. We have a cell culture lab in our medical institute during the last 8 or 10 years. And always we have this from funding this institute, we have microbiological lab It's also should be useful for alternative experiments model. Uh, is now with some experience in practice, it's possible to produce cell cultures from living animals or from tumors. It is this possible due different technique, and if we have some uh, uh, cell culture media, it's not so difficult to produce this. Uh, Another alternative method is creating computer and mathematical models before animal experiments. And of course, these digital models not, eh, I think, impossible to uh, uh, this no, it's not equal for future usage. Some chemicals or approaches in humans without animal testing, because it's, as we remember, production experiments is very important, because in real life it's in 90% is work different from theoretical. And but the calculations use some artificial intelligence or computer programs is can give some very relevant and useful information. Probably nowadays it's more effective than previous calculation without use artificial intelligence. But if in case if we have more strict legislation, it's way for solution. Um, yes, animal-free model have some advantage, but nowadays still scientific journals um, prefer experiments with animals. It's more valuable, and this research more attractive for publication in big journals because it's uh, computer modeling very easy and cheap and very quick, but not as so reliable for this scientific society. Uh, but in this case, we reducing cost for experiments. We increase speed of in decrease time for these uh, experiments, because these animal experiments need a lot of time and efforts and very um, high cost for this activity. Uh, it's interesting, but one mice in Scandinavian country, it's need about 12 crowns or something like that. It's like more $1 per day for this all facilities. And it's in some cases, probably we can compare with human cost living in some countries. It's very strange, but these experiments it's not cost efficient with animals. Okay, it's a key question in this field. Now we move to um, bioethics and PhD research and local practice. What about humans? This is typical. I demonstrate in Ukrainian. Of course, I have some English translation, but I explain. This is document usually we can find in medical cases, medical history form 003 in our hospitals. This these documents it's by default. Patients sign by default, and it's uh, this some very short and undifferent, undefined information, but the main meaning is some collection and this agreement for collection and processing personal data and this some this about some history from of disease is some anamnesis, morbid anamnesis life is also uh, useful in some epidemiological medical research, some statistics, etc. Uh, in this, very simply, it's name, surname, data, and signature. Um, okay, talk about next talk is about definition biosamples. It's any material taken from human bodies, very simply. Uh, usually, usually it's blood, serum, plasma. It's most widespread. It's biosamples. Also, surgery materials, fluids, excretion, and some nuclear acids, which molecules which contain information about human. It's also biosample. Um, yes, I start talking before this lecture. It's like discussion before lecture, but we have a very big gray area of this in field of patients' rights. So, main reason is not not so define boundaries of uh, these patient rights in Ukraine. And two is not so clear for this certain rights. But patient should be informed about research. It's receive all clarification about procedures and some examination about risk and discomfort, about some complication. Uh, Based on all this information, available information, patients should make decision about take take participation or refusing of this study. Also, this information should should provide this information about future uh, usage of these biospecimens for what purposes. An example is creation of diagnostic tools or treatment or methods or prophylaxis methods. Uh, Also, patients should be informed about transfer information to scientific database, about scientific publication, and this um, also usually when patients inform about somebody receive benefit from this biospecimens, and this is usually patients refuse to take agreement to usage these biospecimens. And it's a lot of psychology needs for working with patients. And nowadays, it's we have fast development of biobanks, and uh, this system works in this way. Doctors explain for patients about some uh, big role in humanity development and creation of goods for all mankind. But people who work in biobanking receive direct some privileges or some benefits from this activity. Uh, Due patient participation. It's this problem because it's difficult to it's could be like on commercial variation of this interaction between patient and this research institution. But many research institutions directly not receive so obvious benefit or obvious income. Uh, these benefits received on next participants like pharmacology company or this business, big pharma business, or this uh, company which produce some devices or some structures, private structure. It's uh, lots of problem which is very difficult to solve. This in this situation, scientists uh, receive some benefit, but it's mostly it's non-material. It's like some intermediate stage. It's some justice for this system needs some justice for this distribute benefits from this activity for all participants. But solution is so far. Okay, thank you for your attention.